Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 13 of the series of our online lectures. Today we are going to talk about translation in eukaryotes. Now, in the previous two lectures, we talked about the process of translation in prokaryotes. As you know that there are similarities and differences between the processes of translation in prokaryotes and eukaryotes. So this is one of the main reasons why I decided to talk about this process of translation in prokaryotes and eukaryotes separately. Having studied about the process of translation in prokaryotes, it should be very helpful in understanding the process of eukaryotes, because there are a lot of similarities between the overall process of translation in prokaryotes and eukaryotes, but still I believe that it is much more interesting to study them separately to be able to understand the differences as well. So now we move to the outline of our today's lecture. The outline of today's lecture is we talk about eukaryotic translation. Then we talk about the initiation of translation. We talk about how peptide bond formation takes place in eukaryotes, and then we talk about the process of translocation, and like in prokaryotes, the process of termination of translation also occurs in eukaryotes. So we will talk about the process of translation and the process of termination in eukaryotes. Now we move towards our lecture. In prokaryotic cell, transcription and translation are coupled. That is because, as you know, that in prokaryotes there is no nucleus, no nuclear membrane. So the process of transcription and translation are coupled. That is, when the RNA is transcribed, it is also translated. That is, translation begins while the messenger RNA is still being synthesized. Whereas as compared to prokaryotes, in a eukaryotic cell, transcription occurs in the nucleus. Obviously, nucleus is surrounded by nuclear membrane, and translation occur in the cytoplasm. So, in eukaryotes, the nucleus and the cytoplasm, they are two distinct compartments. So, in eukaryotic cell, transcription occur in the nucleus and translation occur in the cytoplasm. Translation process in eukaryotes involve activation, not essentially the step of translation, like I told you about this in the process of translation in prokaryotes. This occurs the same way as in prokaryotes. Then after activation, there is the process of initiation, elongation, and termination. So you can see that the overall steps in the eukaryotic translation are similar to that of the overall step in prokaryotic translation, but the details of these processes and the different factors that are involved in the process of translation in prokaryotes and eukaryotes, they can be and they are different from each other. We talk about this. Once you study the process of translation in eukaryotes, you will be able to correlate the similarities and differences between the processes in prokaryotes and eukaryotes. Now, initiation. The initiation of translation in eukaryotes is complex, involving at least 10 initiation factors, EIFs, and is divided into 3 steps. First is formation of 43S pre-initiation complex. This is followed by formation of 48S initiation complex, which is followed by formation of 80S initiation complex. Remember, in prokaryotes, the final complex was 70S because the ribosomes in the prokaryotes are smaller as compared to the eukaryotes. So formation of 80S initiation complex takes place. Now, formation of 43S pre-initiation complex. A ternary complex containing MET transfer RNA and EIF2 bound to GTP attaches to 40S ribosomal subunit to form 43S pre-initiation complex. The presence of EIF3 and EIF1A stabilizes this complex. Now, formation of 48S initiation complex. The binding of messenger RNA to 43S pre-initiation complex results in formation of 48S initiation complex. EIF4F is formed by the association of EIF4G, EIF4A with EIF4E. The EIF4F, referred to as cape binding protein, binds to the cape of mRNA or messenger RNA. Then EIF4A and EIFB binds to mRNA and reduces its complex structure. This mRNA is then transferred to 43S complex. The ribosomal initiation complex scans the mRNA for identification of appropriate initiation codon. 5AUG is the initiation codon. Now, formation of 80S initiation complex. 48S initiation complex binds to 60S ribosomal subunit to form 80S initiation complex. The binding involves hydrolysis of GTP bound to EIF2. This step is facilitated by the involvement of EIF5. As the 80S complex is formed, the initiation factors bound to 43S initiation complex are released and recycled. Now, this is the diagrammatic representation of the different factors. You can see here EIF3, this is EIF4A, this is the cape, this is the mRNA, This is EIF4G. This is PABP. This is EIF5. This is mid transfer RNA. This is EIF2. This is the 40S unit, subunit. Now, elongation. Ribosomes elongate the polypeptide chain by sequential addition of amino acids. The amino acid sequence is determined by the order of the codons in the specific messenger RNA. It is very clear, it should be understood that the amino acids, they are not added randomly, but they are added in a sequence that is determined by the codons that are present in that particular messenger RNA. The information is provided by the messenger RNA, and according to those information that are provided by the messenger RNA, the amino acids are added. So this process of which amino acid would be added next depends on the codon sequence of the messenger RNA. Elongation is a cyclic process involving certain elongation factors. A cyclic process means that this process of elongation continues in a cyclic manner, so that the messenger RNA is read and the different elongation factors, they are recycled to be able to add new amino acids according to the codons that are present in the messenger RNA. Elongation may be divided into 3 steps. The first of these 3 steps is binding of amino acyl transfer RNA to A site. Then is the peptide bond formation. Then is the process of translocation. Now, binding of amino acyl transfer RNA to A site. The ATS initiation complex contains MET transfer RNA in the P site and A site is free. Another amino acyl transfer RNA is placed in the A site. Now, once another amino acyl transfer RNA is placed in the A site, we have both the P and A site occupied. This requires proper codon recognition on messenger RNA and involvement of elongation factor 1A and supply of energy by GTP. Because this is an energy requiring step, this is why these processes are needs to be properly controlled, because in terms of energy, overall this process of translation is expensive because GTP is consumed. The amino acyl transfer RNA is placed in the A site. Elongation factor 1A and GDP are recycled to bring another amino acyl transfer RNA. Now is the step of peptide bond formation. The peptidyl transferase catalyzes the formation of peptide bond. Net result of peptide bond formation is the attachment of the growing peptide chain to the transfer RNA in A site. Then comes the stage of translocation. The ribosome moves to the next codon of messenger RNA towards 3' end, because the messenger RNA is read in the direction 5' to 3'. This process is called translocation. It involves the movement of growing peptide chain from A site to P site. Translocation require elongation factor 2 and GTP. GTP get hydrolyzed and supplies energy to move messenger RNA. Elongation factor 2 and GTP complex recycles for translocation. Now this is the stepwise process shown in the diagram. This is the 40S subunit, this is the 60S subunit. Here is the exit site, here is the P site, here is the A site. This is the in the red shown messenger RNA in the 5' to 3' direction. This is the incoming amino acid transfer RNA with the other factors. In the step 1 decoding, you can see here the two sites occupied. Then here is the bond formation that is taking place, and then peptidyl transferase in step 2, peptide bond formation it takes place, and here is the process of translocation. The machinery translocate, and the A site is now empty, and through the E or the exit, the transfer RNA which brought the amino acid is released. Now ribosome waiting for next amino acid transfer RNA. Now in the next step, because we talked about this, it is a cyclic process, another amino acid transfer RNA will come and will occupy the A site. Once again the peptide bond formation will take place, and once again then the process of translocation will take place. Again the A site will become empty, and in the coming in the next step, the incoming amino acid transfer RNA again will bring a new amino acid according to the codon sequence that is provided by the messenger RNA. And in this way, this elongation cycle will continue. It will continue until when? Until the stop codon, until the translation machinery encounters stop codon. Once the translation machinery encounters stop codon, further elongation of the chain of the newly formed polypeptide will stop, and then the process of termination will begin. So this is how overall the process of elongation in eukaryotes takes place. Now once this process of elongation the elongation cycle encounter one of the stop signals, UGA, UAA or UAG. Again mistake, sorry about this. So UGA, UA or UAG terminates the growing polypeptide. When the ribosome encounters a stop codon, there is no transfer RNA available to bind to the A site of ribosome. Instead, a release factor binds to it. In eukaryotes, ERF1, eukaryotic release factor 1, recognizes all the three stop codon, and eukaryotic release factor 3 stimulate the termination events. Now, once the release factor binds the ribosome, unit falls apart, releasing the large and small subunits. The transfer RNA carrying the polypeptide is also released, freeing up the polypeptide product. Now this is how you can see, this is the cap, this is the messenger RNA, these are the release factor, eukaryotic release factor 1, eukaryotic release factor 3, this is the GTP, this is the stop codon, and this is how the 40S and 60S subunits, they are dissociated. Release factors also release, chain of amino acids, the newly formed polypeptide is released. Now this is the end of the process of translation in eukaryotes. Thank you for watching. If you have any question, please feel free to contact me. Thank you.